Hello, thanks for being here, guys. Welcome to the Natural Science or to the STEM Success Seminar series. It's my pleasure today to introduce one of my former students and a Scott Science Scholar, Maryville College alumnus, Boomer Russell. So Boomer graduated 2019, is that right? Was a member of the class of Scott Science Scholars, I guess that started in 2015. While Boomer was here, was involved in many activities. So Scott Science Scholar, played tennis, was a member of the student programming board, was a Scott Science Scholar peer mentor, was a Maryville College ambassador, had an exemplary senior study, did undergraduate research in my lab starting, sophomore year? Is that when you started?
It was sophomore year.
Yeah, sophomore year all the way through. Did an internship at UT during the summer, working on plant receptors. Okay, plant receptors, and had a presentation at a major science meeting, all while he was an undergrad. He since moved to the University of Tennessee, the Department of Biochemistry, Cell and Molecular Biology, and has three papers already published as a fourth-year grad student, which is amazing. Two of those are first author papers, and also two of them are in the Journal of Biological Chemistry, which I think is funny because Boomer always said seemed to be maybe a little bit math phobic, and in order to publish in JBC,
I might still be.
You got to have the math chop. So it's interesting that he's kind of digging hardcore into physical biochemistry, and so love that. But he got some good training on the fluorimeter in my lab, and so hopefully built up his endurance and stamina at the very least. At UT, as a grad student, Boomer has also been the recipient of the Gate Fellowship, the Robert and Nell Keenan Bioscience Fellowship. He had a travel award from the Biophysical Society meeting, and is a recipient of the Dr. Cynthia Peterson Fellowship, which is named after my graduate school PI. So very exciting and a nice little circle of life to see those connections. Anyway, Boomer's going to tell you about his work and his plans for the future, and probably a little bit about his path to this point. And so, welcome, Boomer Russell.
Thank you, Dr. Gibson. Hi, everybody. How are we all doing today?
Good.
I'm glad that's one of you. Great. If you got one thing from Dr. Gibson's introduction that you should know about me, I'm tired and bad at math. Okay. But today I'm going to tell you about the work that I've been doing at the University of Tennessee. I'm going to try to keep it as less boring as possible. You're welcome to interrupt me with questions, but then more importantly, I want to make sure I talk about my journey, my experience, things like that, because I know that's what you all are here for. So the title of my talk today is the Candida albicans virulence factor, candidalysin, polymerizes in solution to form membrane pores and damage epithelial cells. Before I get into that, I would go into these slides about myself, but Dr. Gibson did a very good job at explaining all of those things. So I'm just going to highlight this very cute picture of us from when I graduated back in 2019. And as she emphasized, I was heavily over involved and very grateful for it. Did many things, and I purposely included this picture because we're here again. It's really weird how things work like that. See, and like she said, I'm at the University of Tennessee. I work for Dr. Francisco Barrera. And our department is very diverse. You know, there's a lot of keywords in there: biochemistry, cellular, and molecular biology. So that covers a lot of aspects of natural sciences, particularly at the molecular level. I specifically, I guess, if I were to pick my niche area of interest, would be membrane protein biophysics, which is incredibly ironic because I did very bad at regular physics while I was here at Maryville. I also did very bad at chemistry, which is also really ironic. But I also did fairly decent at biochemistry and really liked it. And so that's kind of what led me to here today. And so, like I said, this is what I'm going to talk about. And so the important player in this that I want to introduce you to is Candida albicans, which is a fungal pathogen that typically exists as a commensal fungus, harmless, inside of all of you right now. It's important for maintaining your immunity and your microbial immune stimulation, things like that. But the part that becomes interesting is when it transitions into this pathogenic hyphal state. It grows this long hyphae and can cause a lot of havoc to your body. More notably, Candida albicans infections of the mouth, or thrush, more commonly known, is one of the most common symptoms of untreated HIV. Vulvovaginal candidiasis affects 75% of all women in their lifetime. And then systemic candidiasis, if it transfers the blood barrier and gets into other organs, has a 50% mortality rate, which is very scary and not good, which is the hook that I'm using to reel you in to make you interested in my talk and why you should care. And so you think that the goal of my work would be to develop effective biosensing and therapeutic avenues to treat these infections. Well, you might be saying to yourself, but boomer, antifungal treatments exist. And whether or not you're saying it to yourself, or if it's just me putting it into your brain, I'm going to say it to you today. And the reason I'm saying that is because I'm going to argue against it. Antifungal treatment costs a lot of money. Typically, not just because you have to take an antifungal, but usually that comes with some antibiotics as well. We are also seeing a large increase in drug resistance among different strains of fungal pathogens. More on the public health side of these things, whether you know or not, our country has an effective, fairly marginalized healthcare. So for certain populations that are typically targeted by these infections, they have a hard time getting treatment. And then more importantly, the way that these studies are carried out, there are many gender disparities as they lack to understand the nuance of, for example, like trans men, trans women, et cetera, that can also be affected by these by this disease. And so I would like to change the statement instead, developing effective biosensing and therapeutic strategies to treat Candida albicans infections is an intersectional global public health concern. And I'm going to talk about a little player that is important in this. And that's this little peptide, 31 amino acids, and its name is candidalysin. Candida for the species it comes from, lysin just meaning that it cuts things. And I'll tell you a little bit about why that is later. But recently in the literature, from what I read when I started and joined my lab, it was kind of raising headlines for being this really important virulence factor to cause infections. It was discovered and known to be required for Candida albicans infection. So if you knock out the gene that goes to this protein, the Candida albicans cannot infect human cells. And so it was obviously deemed very important. So people started to try and learn about it. People learned a lot about the immune signaling it causes, what happens when it activates different signaling cascades, things like that. But nobody actually thought to look at how it actually did all these things. They learned all the outputs, but not actually the mechanism of why that happened. And so that's kind of where I came in. When I originally made this presentation at the time, my first author paper was on bioRxiv. That's just a server that allows you to place your preprints before they become published. But now, instead of it being under revision at eLife, it is now published and out for the public, which has been a very cool experience. And if you'd like to learn what it is like to publish a paper, I'd be very happy to talk about it later. But I'm going to get to it with the results of what we found. And so candidalysin is a pore-forming peptide that undergoes a unique mechanism of self-assembly to form pores and cell membranes and cause cellular toxicity. And so I'm going to go through each part of this. The first being its ability to form these complex assemblies. And just like if you work with Dr. Gibson, or if you've taken biochemistry and done biochemistry lab, you probably run an SDS page gel. And what all biochemists do when they start a project, they throw that thing on a gel and they see what comes out. And so we did that, and we like we expected, saw the monomer, the single subunit at its molecular weight. However, we were surprised to see this higher molecular weight species here at about 26, because that's very uncommon for proteins that undergo this treatment process to maintain any type of larger assembly. The size that corresponds to is an octamer, just by our crude calculations. Not bad math. But we took that and we ran with it because it seemed that this was incredibly interesting. And so we went through, I won't go through the nitty gritty of these things, but we performed a lot of different experiments that essentially allow you to measure the sizes of proteins. And what we found is that candidalysin is able to oligomerize without lipids. And that's really weird. And I'll tell you why. It's not weird to you right now, but it's about to be, I promise. Peptides like candidalysin typically require binding to a membrane to do any type of self-assembly. Raise your hand if you've ever been stung by a bee. Good, a lot of you. The thing in the bee venom that causes the pain and swelling is a small pore-forming peptide called melittin. When melittin is injected onto your skin cells, it binds to the membrane, forms these assemblies, and eventually just tears the membrane apart. And that hurts. That's why they hurt. Because they're literally poking holes in your cells when you get stung by a bee. But candidalysin does something similar, just like a little different. And so we did these very advanced, super fancy biochemical techniques, stuff, to find out that candidalysin has this ability to form these large molecular weight species in solution, just like by itself, which is just super quirky and really weird. This doesn't usually happen. Candidalysin is really small. Its ability to have domains that allow it to bind to itself would be very unlikely, but obviously it is very efficient at forming these large species. And our next question was, what do they look like? And so we have some collaborators that we work with at the University of Missouri that do this really cool technique called atomic force microscopy. If you think of any type of microscopy, you're taking a sample and you're looking at it under a lens with some light. Think of this as you have your sample and you're running essentially a cantilever over it, and it's measuring different heights of your sample relative to the sample that it's on. And so you're able to get these images that show you physical shapes of what's going on. And what we were really intrigued to find is that candidalysin can form what almost seems to be these like stepwise growing linear polymers that eventually close to form loops. And that's just weird. I mean, there's not really anything if ands or buts about it. It's just weird, so we wanted to learn more about it. Especially considering the role of this peptide is to cause damage. And so we wanted to go into the next part of this mechanism, which was its pore-forming ability. And so, using the same technique, atomic force microscopy, you take your sample slide and you essentially put a membrane on top of it. And then you can treat that membrane with your sample. And so we treated it with our peptide, and we were able to observe different pore species. So you see over time, when you treat with your peptide, you get these different populations of pores. But what was really weird is that we saw different kinds of pores. We saw two in particular. One in particular, we call unrimmed, as you see here. And then we call the other one rimmed. You can see these white specks about the periphery, and I'll get into what those are in a second. And so what we thought, we wondered if our loops that we saw in solution were similar to our pores. And that's exactly what we saw. When you do this experiment, you can wash away, when you like wash your hands or you do your dishes, you can use a detergent to wash away all those oils. You can do the same thing in this technique, just a bit more controlled. And when you wash away the bilayer, the membrane that's here, you can see where there were pores, as pointed by these blue arrows. There are these loop structures. And that was incredibly convenient to our hypothesis, because also the sizes of these loops that we saw in solution matched the size and area of our unrimmed pores. And what that made us think is that these loops insert into the membrane as unrimmed pores through this almost like dynamic association-dissociation process, and eventually stabilize into this smaller rimmed pore. And so we wanted to experimentally connect the self-assembly that we saw to pore formation. And so to do that, we did a mutational analysis of our peptide, did a screening, and we found these two variants. That one was a gain of function in green, or called I9A, and one is a loss of function in red, or G4W. This just refers to the amino acids that were replaced and substituted that caused mutations. And we did some experiments to see how they changed, or how they compared to wild type seen here. And we were very happy to see that our gain of function variant was able to very efficiently cause membrane damage. This graph is from the results of, I call it my bread and butter assay. It is a fluorescent dye release assay that can just measure membrane disruption over time as an output of fluorescence. And so you have lipid vesicles filled with an environmentally sensitive dye. You treat with your pore-forming peptide, and the release of that dye, you can measure it over time. And the greater the amount of fluorescence, the greater the leakage. And so what we saw in comparison to wild type, that our gain of function was very good at it. And our loss of function was absolutely awful. Didn't get the memo, didn't understand. And we did some of our techniques that we used to study its self-assembly properties. We saw the same thing. Our gain of function was very good at forming these larger polymers, whereas our loss of function variant was able to form something, but not in high enough abundance and likely not efficiently enough to form a pore. And then even when you look at it under an atomic force microscope, we see the same occurrence. We see more self-assemblies in our gain of function and no assemblies in our loss of function. The really cool thing that we saw is that in here, if you notice, we see a higher abundance of loops, which if our hypothesis is correct, loops equal pores eventually. And so if it is more efficient at forming loops, then it is also more efficient at forming pores. whereas our loss of function variant did not do that at all. You can see some specks, so it's able to kind of aggregate, come together, probably precipitate out of solution, but not enough to actually form a pore and cause damage. And we did some cellular assays, some Western blots, treating with some mammalian epithelial cells, and looked for biomarkers of Candida albicans infections. And we see those same effects are recapitulated in epithelial cells as well, in which these two different markers, in the presence of our gain and loss of function variants, and the presence of our gain of function, they are in higher abundance, meaning that it's likely more effective and more damage prone, whereas it is heavily reduced in the presence of our loss of function variants, which led us to publishing papers to tell the world about this mechanism, this really unique mechanism of pore formation, in which candidalysin is secreted from the fungus as a monomer, or likely as a monomer, undergoes a chaotic yet somewhat kind of specific self-assembly process to form this oligomer that serves as the site for polymerization. Think of this kind of like those markers you used to play with as a kid. They would stack on top of each other and like hit your little brother with. They kind of come together as a chain, and then eventually they're flexible enough to close in on themselves and to form a loop. And then those loops are, we call them membrane competent, and are able to actually interact and bind to membrane lipids that undergo this conformational switch in the membrane from this horizontally oriented structure into a vertical one. Because what that would do, that switch would allow these white spots we hypothesized are proteins. And so that would allow for it to be more stable in the membrane and just cause greater damage. A lot of the experiments that we do are in vitro, just like experimental, very synthetic, not actually in a cell. And if you think about the environment in which these yeast infections occur, I mean, you have these tiny channels that the fungi are digging into your epithelium and then releasing this product in these very complex spaces with complex lipids, different proteins. And so we're very far from fully understanding what truly is happening, but this is the first time we're able to get I'll say a surface level idea of what is likely going on. And so since we published all this, well, it was published in 2022. We first submitted it in 2021, did some revisions, whole process. We can talk about that later if you want. Then wrote a review about it too, combining all the information together. But since then, the fabulous part of science is when you get to make a nice, pretty little picture and communicate your science to people at conferences and things like this, right? The really ugly part is that like it's been about a year and we've just had a hard time going from that and figuring out what's happening next, because this process in particular is incredibly dynamic and really complicated. We're starting to grasp at some straws and we finally got something. But I mean, if you're a senior or a junior and you're doing research, or if you're doing your senior study, that's just kind of what science is like. Sometimes it's really pretty and it works really well and it just fits your story. But most of the time it just doesn't. And it's really important for you to learn how to persevere and to critically think and try to figure out what is happening. So that's all I have to talk about with my research. I figured I would end my talk with some, I'm going to call them some fun facts and tidbits of wisdom, because as Dr. Gibson summarized, I'm old, tired, and bad at math. And so we're going to go over all three of those things. Not really, just maybe the tired part. I graduated from Maryville College in 2019, and I went straight into grad school. I was really lucky. I applied to 5 or 6 different programs, got one interview, and really got into that school and ran with it. Sometimes you just have to take what you get and go, and frankly, it's worked out really well. I love my department. I love what I get to work with, and I think I'm very lucky and fortunate to have had that experience. Here's some things I've learned. My grad school experience has been very interesting, as we've experienced some very silly little situations in the world since 2019. And so it really makes you learn, grad school makes you learn. And so these are some things that I guess I'll go over and we can talk about more philosophically later. I'm also going to put my email on the PowerPoint at the end of the evening or at the end of the presentation. Celebrate your identity and who you are. I will stand by the fact I came to college in the closet. I was not confident. I was not allowing myself to, you can ask Dr. Gibson, she's my advisor. I was really bad at most subjects. Grades were not working. Science was not going well. But then I changed that. I came to a realization. And after that happened, I started to let myself be smart. Before that happened, I was just like this little fat kid that made jokes about getting C's. And then when I came out, I was this little fat kid that made A minuses, which is very helpful for this, and it also helped me find what I really enjoyed. Dr. Gibson listed off a bunch of things that I did in undergrad, and I am grateful for all of them. They really helped me kind of figure out what I wanted to do. You know, I came in like most science suckers, and I was pre-med biology. I was gonna go to medical school, and all the things. And then I made a C+ in Gen Chem, and really just changed my worldview. Not for a while though. I was pretty on the pre-med train for a while. But then I started doing work with Dr. Gibson, as she so kindly, for some reason, let me do. I was not qualified. And doing research, being able to critically think, and her allowing me to have the freedom to gain the confidence to figure these problems out was very helpful for my self-realization that I wanted to do research. I wanted to go to graduate school. But I think science is so cool. I think it deserves to be, everybody deserves to have access to it. I think the pandemic really showed that we need to work on our science communication and access, and make ourselves people who educate and teach people and make science a very all-inclusive kind of field. I mean, even more recently, as the pandemic has slowed down, I suppose, I started doing drag. I'm a drag queen at night. You can call me Fatty Acid. It's very funny like that. But that is very much a part of my identity now. And I love doing that. It is a major part of what I do. It's a great release from graduate school. It's very different from graduate school. But it is one of the best things that I've gotten to do. But it's all together in I used to do this. I gave a couple talks when I was younger about STEM identity. And this is, I think Dr. Gibson or Dr. Siopsis have done the same thing. And it's very much a part of that because it's all encompassing. This is the part that I wish Dr. Siopsis was here for, because so y'all probably have imposter syndrome. At least you're familiar with it. You've met her before. But she's gonna be around for a long time. No matter like what you do, how well you become at something, like you're always gonna think, I mean, unless like you're insane, but you're always gonna think that somebody's going to realize that you are not who you say you are, or you're not as intelligent, not as talented, whatever. This is a picture from when I was doing the rope tower during my SQ orientation. It was like day 4 or day 5 with all these strangers in this really weird new place. And Dr. Gibson on the ground being like, yeah, you know, that kind of stuff. And I remember so vividly, it took me like an hour and a half to climb the tower. But like, I don't know why I didn't just give up, but I was just thinking, for one, I picked like the hardest side, which was stupid. It's one like the tower and those things anyway. But I just remember so vividly Dr. Siopsis just like being at the bottom, like supporting me, giving me guidance. And then I can't remember if it was a few months later, or I don't know what happened, but she sent me an email or we talked about it and she showed me this picture. Which I was like, how dare you? It's not a cute picture at all. I'm so uncomfortable. But like I think I don't know, I did something, Jess. But I had to force myself to know that I could do this. Even something so simple as climbing the tower, you know, but like it translates to things like getting an A on your exam, or like doing well at like a talent show or something like that. And I mean, this is me after I passed my candidacy exam last year, which was an experience. We can talk about that later also if you want to learn about the ins and outs of graduate school and getting a PhD in science. But it's just something that like the moment that you realize that she's going to be here, imposter syndrome is the moment you can figure out ways to deal with her. And the last thing I want to leave you with is that as you go through school or just you grow up, you know, you start college at 18 and you think like, oh man, wow, look at this world I'm going into. I get my own twin bed. I get to buy a storage ottoman. I'm going to buy all of the ramen specific microwavable bowls that I can. And you're just so excited about it all. And then you graduate and then everything doesn't change necessarily, but you're going to grow up. You're going to meet new people, do new things. You're going to realize your priorities are going to change. And that's something I've really experienced in the past year and a half. I always say that my early 20s were taken away from me by insecurity and the pandemic, because they were, because I had to stay inside and I was insecure. But like after that, in the past like year and a half since it's ended, like I've gotten closer to my community. Like my friendships have gotten stronger with the people that I knew in undergrad, as like two of the suckers that I went to undergrad with are here right now. those friendships get stronger. You meet new people and you learn more things about yourself, and you just like, you find a community, and it changes your priorities. I mean, Dr. Gibson asked me when I got here, what's next? And I'm kind of on the brink of graduation. Like, there is a small light at the end of a very small tunnel. And I don't really know. I mean, I went into grad school very gun-ho thinking I'm going to be a professor. That was like my idea. I basically want to be Dr. Gibson when I grow up. And I still do. I still want to be Dr. Gibson when I grow up. But now that the good thing about a PhD is that it's a very flexible field. There's a lot of nuances. You learn things about like the way you want to live, who you want to talk to, like just stuff like that. So there's so many options, and that all changes over time. And it's okay, you know, sometimes when you're a sophomore in college, you might think, I don't know what I want to do with my life. Well, let me tell you, you could be a fourth year PhD candidate and you can still say, I don't know what I want to do with my life. You have an idea. I feel very fortunate to have known that I wanted to get a PhD, because that's a big decision. Anything after college is a really big decision, frankly. I feel very fortunate that I have the guidance and ability to make those choices. But I just wanted to make sure everybody knew that, like, you're allowed to change your mind. You're allowed to figure something out and feel it out and see if you like it and if it's what you want to do. And so that, I'm going to end my little spiel. I've included my email on the top. Dr. Gibson can also give you one of my emails, and then my school email or that email. I'd love to take any of your questions. It can be about science, it can be about like other things out here. Whatever works.
Do you think you could introduce your gene changes to any albicans into like the wild type population to make it less effective?
Yes. We do not do that. So we are a very biochemistry, biophysics lab. So we don't actually work with any fungus. When I tell people about my research, a lot of them think I'm a microbiologist, and I'm a farthest from that. But we have collaborators who actually do like mouse work. And mice and zebrafish are two very common animal models to study like yeast infections and albicans infections. And so I believe that what's happening, we're going to introduce those mutations into a strain of Candida albicans and then treat mice with them to see if those effects were the same. My assumption is that if I have not heard anything about it, that it did not work. But yeah, so that is exactly like what would be a next step. This is and that has been done in there are other variants of candidalysin that we're learning. And that has also been done where they've seen that some are more toxic or less toxic. Like that. Good question. And also, if you don't ask here, I mean, it's a big room, big scary man at the front. You're welcome to ask another time too. That's really fun. Yes.
Do you have crystallography data or
So-
anything on the pores?
Oh man. So no, we are actually getting ready to do some cryo-EM on it. So the structure itself, let me go back to it. I'll switch to this, just the shape. So actually, good. So this, I'll go through some of these. This in particular is the most useful. This is a spectrum from an experiment called ion mobility mass spectrometry. I didn't do it, collaborators that do it, but it is a type of mass spec. I think there used to be like I thought there was a mass spec machine. No, it is a machine that essentially can measure the sizes of different protein complexes, but it can maintain a oligomeric size. So it measures on the basis of like size and shape, so like number and then like radius and things like that. And so it's very hard to get a pinpoint of what the structure is of this protein because, well, one, it's a peptide. Peptides are usually just like, they're really silly because they're really short, they're really flexible, and they are incredibly dynamic. And what we've learned in just some really basic experiments really is that it's really, when you do experiments that are meant to like give you an insight on what the structure is, like crystallography or cryo-EM or NMR, depending on what the technique is. Like for crystallography, like you need like one thing, like a monomer, a like single subunit of something. You need to form crystals and then do the X-ray diffraction, things like that. Cryo-EM is a bit more fluid, and because this all occurs in a fluid environment, it allows you to get a better idea of like snapshots of different like orientations. Because I mean, here I have it very drawn out in this really cute cartoon. But like in reality, this peptide is going to be like all which ways, all directions. And so figuring out one, what's going on, but also if we can find conditions that promotes the assembly of a certain species, so like of a particular like oligomer or like a loop, that would be how we could go about doing that. Or just conditions too. We have some like data saying that when you put the peptide in a buffer versus just water, it behaves differently. Makes things incredibly complicated. But yes, we want to do that, but there is no like structural data. They were able to make a model from this based on a different sequence. That's how we got the octamer in this particular orientation. And there's some simulations you can do with AFM data to give you general orientation and things and length and flexibility, but other than that, that's it for now. It'd be nice.
Yeah.
Really super cool.
It's nice though that you know with the ATF data that it's so infrequent that we have these biological models, molecular models that you can actually see physical evidence that emulates what actually is the shape of what's going on biologically.
Oh yeah, this is like at like the nanometer level, like that's tiny. And it's been super helpful. And AFM is a very common technique in the field of like membrane active peptides broadly, especially ones that form pores. But yeah, very neat, very cool. I can't remember. I saw your hand go up.
Yes. I was wondering what your day-to-day looks like in grad school and if it's very different from your first year.
Oh, incredibly different. So grad school, I consider grad school, especially after so first year is very undergrad-ish. That's when you take the most classes. The priority of getting a PhD in science is to do the research and contribute your knowledge to a field, less so like taking exams. So I really only took like true exams like my first semester. We have like kind of like our lead out course per se, I guess. There's like advanced protein biochemistry, cell biology, and then you just have a bunch of other things. But in your first year, you're taking courses. You're also teaching. So it depends on the school, but at UT, your schooling is funded by until you're in a lab, is funded by your being a teaching assistant. And so in graduate school, I'll start here. You don't pay for it. You should not pay for a science PhD. You get a tuition waiver, a stipend. It's supposed to be a livable stipend. And our stipend has gone up, actually. Our stipend just went up to 26,000 this past year, or it's going to go up to 26, with the goal of eventually getting to 30. And then you get health insurance. And so it's, in that sense, it's like a job. But when you start, you're a teaching assistant. And so you like TA a course. So like, fairly similar to like what your like biochem SI or organic chem SI leader would do, kind of in that same realm. Some of them like teach additional lecture courses, some of them like run a lab section, things like that. So your first year is like courses, teaching, but you in the program in hours, we rotated in different labs. We rotated in 3 labs for 6 weeks each, just so we get a taste of each one to see where we wanted to settle. And also to like get them to want to take us. After the first year, they kind of your course load goes down. You do credit hours you have to take, but it is significantly less. And I consider grad school more of like a job, more like work. Like I go into the lab, and it's the nice part is that it's incredibly flexible. Our lab is we're very 9 to 5. We don't like to take our work home with us as much as we don't have to. That kind of stuff. There are some labs that are like 7:30 in the morning to like 7 at night. There's some labs that can like 11 a.m. and stay late. So you can be really flexible. A lot of that depends on the lab culture, which is always something to consider when you are looking to join a lab in graduate school. But my day-to-day, since that was your question, it's a very long way to get to that point. I go to lab like 9 o'clock is when I like to get in on a good day. And I'll usually stay until like maybe 4 o'clock. But it just depends on what I have to do that day. Some days I leave early because I'm done with my work. Our lab is very, get your work done and you're fine. Sometimes if I have a lot of writing or even reading to do, I'll go to a coffee shop to work. Not all labs are like that. My PI, my boss is very kind and is okay with that as long as we get our work done. Some professors are very like, I have to see you in the lab working or else I don't think you're working. And so those are things that you can consider when you talk to other people about. But my day-to-day is pretty, I'm not going to work today because I'm doing this. And it's because it can be that kind of flexible. So, did I answer your question?
Yes, thank you.
Oh, and I will say, I just want to point this out. I'm sure you've heard a lot of like horror stories about graduate school, and like those do happen. I have learned that a lot of that is also very, it's a good hot take, it's self-inflicted. The pain that a lot of grad students like harbor onto themselves can not always, but can be very self-inflicted. Like you can manage your time, you can set boundaries, and some of those are really hard to do, but you can do that. Like I don't work on weekends. I hate working on weekends. I like to sleep in on weekends. But like you don't have to do those. I don't like staying at labs super late unless like I have to. You know, like sure, sometimes there's an experiment that lasts some time. It depends on the system you work in, like all these things, but like you can set boundaries and you can really shape your experience the way you want it to be. So just want to make sure that's very clear. That's a smith, unless it's just scratching the things out.
Along with that, could you speak to some tips or maybe some ways that you think are helpful to endear yourself to your lab mates or become a better like functioning member of a lab? Like what would you want to see if someone came into your lab now with your, for your
That's a great question. And so every year we have first year students come in to rotate for those 6 weeks like I talked about. And what I always look for, there's a couple of things. I mean, in my opinion, when you are doing a lab rotation to see where you're gonna essentially live for the next like, I don't know, 6 years. So it's a long time. You're gonna work with those people, that box for a long time. So and it's also for me, it's like I am seeing who I could potentially work with. And so I take that very seriously and very intentionally. And so I look for people who are very self-motivated, who ask questions and are teachable. It's very like gonna be very dramatic. I'm dumb as rocks. Like absolutely not that smart. I am, but also like I can't really teachable. That's probably like one of my greatest assets is that like I listen, I take notes, and I learn. But also I'm not afraid to ask questions. I know it's hard to ask questions because there are some grad students who don't make themselves that friendly to ask questions to, and that's a red flag you should keep in mind whenever you're doing those things. But no, you should. I look for people who are asking questions, who are taking initiative, who seem excited about the work. I mean, if I know you're just like testing out for a couple of weeks and you have to learn something else new, but in general, like you should come in like fairly eager, like ready to learn, like that kind of stuff. I think if there are other things. Talk to us. Again, we are literally just like we're a group of people interacting together. Like the people I work with every day, they're like some of my best friends. Like I got so lucky. And so we eat lunch together every day, like because at the end of the day, it's like sure your boss is the final say who joins your lab, but like they get input from the students. So it's like if they don't talk to us, I'm not gonna know anything about you. And I'm probably not gonna like you either. So talk to us, ask questions, be initiative, like have some like self-ambition kind of stuff. So were those helpful ones? Great. I'm sure there's more that I can think of. Those ones are just on that topic. Yes.
Talked about making imposter syndrome your friend. You have tips on how to do that, or what does that mean?
Yeah.
Other than just sort of recognizing it's always going to be there, which is important.
Yeah.
Right?
Part of it's just recognizing it and just like, what's the word? The word right now, but just embracing that it's there. At some point, I mean, so in a PhD, typically you have to go through some form of like candidacy. Some programs call it qualifying exams, quals. In a master's, some thesis, defense, whatever. Even when I did my senior thesis here, when you're doing any type of research, you become the expert on that topic. Like I was the expert on TLT1 binding to fibrinogen when I worked here, or when I went to school here. I didn't work here. I learned here. And that's really scary to do when there are people around you who are authorities on those, or who just have done it longer, or know more about it, right? Or people who just like purposely, what you'll find in science, there are people who purposely want to throw you off. They're not super great people all the time. There's a lot of really kind people. I've met more kind people than not kind people, which I think is very lucky. But I mean, with regards to dealing with imposter syndrome, embracing it for one, like you have to acknowledge that you are not feeling confident. And that really is a hard thing to do. But then you eventually like, it's really hard, but it takes a lot of self-reflect. A lot. Like self-reflect and like realize what you've done. And like that you have the ability to do something. I'm trying to think of a really good example. I mean, even when I took biochemistry here, you know, I came into Dr. Gibson biochemistry class straight off of a hot C in organic chemistry 2. You know, semester 1 I got a 74, semester 2 I made a 75. I had momentum, linear rise, right? But no, I was terrified because like, I'm a junior and I'm not doing super great at these subjects that I think I want to do for the rest of my life. What's about to happen? And so I just like really like I started to study. I mean, I studied a lot of organic, frankly. I think I did very fine to study a lot of organic. Organic's just like and Dr. Hughes is also lit as a teacher. But like I just remember like it clicked for me. So like for me, biochemistry was it. Like that was like the thing, right? For some people it is organic. Organic is like the thing, which is so weird to me. It's so awful. But like you have to prepare yourself, you know. I studied a lot for biochemistry. I enjoyed it. I really liked drawing glycolysis over and over on the chalkboards in thaw, and like just drawing or like writing down amino acid names and like drawing the structures of it. That's not a joke. I actually enjoyed doing that. I don't know why, but part of it is preparation. So like if you're scared that you don't have the ability to do something, work on it, you know. And eventually when you work on something long enough and well enough, people are going to praise you for it. And I am the authority of not taking compliments. I hate when people compliment me, or like I used to hate it a lot. Like I just like, I'd make a side comment or I'd like laugh. Like, and I work really hard now to not do that. Because one, it's like a little insulting, because they're saying something nice to you. But also like, take the compliment. Like there's a Vine where there's the face thing, it's like take the compliment, you know, that thing. But like take it and like embrace it too. Like you're allowed to like be confident. My best friend from undergrad tells me I have a huge ego. And she is correct. And I used to be offended by that. And you're allowed to have a huge ego. Just like back it up and believe that you have it. I mean, you're a junior, a sophomore in college, a senior in college, you're about to graduate. Like you did that. Like it's all very blurred when you're going through it. And it's like, oh, I'm like dragging along, getting through this thing, this really hard thing to do. But like you did that, girl. Like you made it through. Or like if you're doing research and if I got the fluorimeter to turn on, that was a big deal. I was very excited. But no, it's like you have to reflect. You have to be honest with yourself. And you have to put in the work. Like being confident about something involves work. Unless you're like full on delusional, and that's okay. You need some amount of delusion, I think, to be a graduate student, a successful one. So you have to think like you are the smartest person in the room. Like you are the authority of whatever you're talking about. Because you're studying it for like I don't know, 8 hours a day, 5 days a week. Like, and that can be applied to anything else that you're doing as well. Is that helpful?
Yes.
I feel like that was helpful. It's very hard to do those things. So don't be hard. Also, don't be hard on yourself. Give yourself like a bit of slack. It's like a little bit.
Good questions? Well, if not, just a couple of other announcements. First of all, thank you, Boone.
Thank you for having me.
I'm sure that you'll be happy to talk about
Oh,
if they want to come
absolutely.
afterwards.